Next, we'll have Ripan Malhi from the University of Illinois at Urbana-Champaign.
Good afternoon, everyone. First, I'd like to pay my respects to the First Nations of peoples of this land, and also thank the organizers of the symposium. It's been a great day of talks. I've already learned a lot, and I'm looking forward to the workshop over the next 2 days. So today, I want to talk about partnerships with First Nations of British Columbia on studies that include DNA analysis that I do with, I work with Dr. Jerome Sobolski on this. Dr. Sobolski couldn't be here today, but he sends his regards. So let's see, I want to start by talking about our partnership principles, and these are respect, mutual benefit, and communication. These principles should look familiar as they are the foundation of any healthy relationship. They also overlap with the ethical principles of the Belmont Report. So as I proceed through this talk, I will provide examples of how we operationalize these principles, as well as employ other safeguards put in place to protect community members and communities beyond the usual expectations of getting IRB approval or Internal Review Board approval. Much of the foundation for these partnerships with First Nations in British Columbia is based on the relationships that Dr. Jerry Sobolski already built when we began DNA studies with these communities. And so here is a picture of Jerry Sobolski with one of our community partners, Harold Harry, with the Canoe Creek Band. So a partnership began with the Canoe Creek Band and nearby communities when Dr. Sobolski visited the community in 2002 to seek authorization and cooperation with the analysis of burials that were recovered nearly 20 years earlier at nearby China Lake. And so this is a photo of China Lake and the nearby area. Jerry gave a talk to community members that was well received, and then the community members took him to other suspected burial sites in their territory where remains were eroding out of the ground. At the request of community members, Jerry was able to confirm that some of these remains were human, specifically at the Big Bar burial site. Over the next few months, community members pondered how to best keep the human remains from further erosion and vandalism, as there was a road nearby that was a high traffic area as it led to a provincial park. Eventually, community members expressed an interest in working with Dr. Sobolski and an archaeologist, Dr. Alan McMillan, to excavate the human remains near Big Bar Lake. The next year, the excavation began with a smudging ceremony. Many community members camped overnight during the excavation to make sure that the remains were not stolen or vandalized. Community members assisted with the excavation or just observed the excavation, and upon completion, additional ceremonies were performed, including a burnt food offering. The First Nation communities agreed to allow limited destructive analysis to obtain information on things like radiocarbon dates, isotope analysis for better understanding diet, and DNA analysis to learn from their ancestors. It was at this point in time that Dr. Sobolski contacted me to help work with him on the DNA studies. With Dr. Sobolski and Dr. McMillan, I visited the First Nation communities in 2005 and gave a public presentation on the uses and limitations of DNA analysis with ancient skeletal remains, as well as with living community members from taking saliva samples. Through the partnership, we established a research study to use DNA as a tool to learn about the population history of the community, in combination with other forms of information like oral histories, traditional knowledge, archaeology, and other forms of information. Protocols were established to anonymize samples taken from living community members and restrict data access to research only on demographic history. Policies were also established to work closely with the communities before publishing any results of the study to safeguard against any potential harms to the community. The China Lake and Big Bar ancestral remains were reburied in 2006 and 2008, respectively. All of these remains dated to about the mid-Holocene, around 5,000 years before present. We were able to analyze mitochondrial DNA at the time of the human remains, and the Big Bar ancestral remains exhibited a mitochondrial DNA lineage that's currently found in individuals that are found on the coast, as well as, I should say, indigenous individuals that are found on the coast, as well as the interior of British Columbia that belong to haplogroup A2. However, the China Lake individuals exhibited a mitochondrial genome that is not currently identified in sampled indigenous individuals from the Americas. So this lineage may either be in really, really low frequency, or it may not exist anymore, possibly as a result of the effects of European contact and colonization. So I just talked about mitochondrial DNA analysis, and so I think what would be useful would be to discuss some of the characteristics of mitochondrial DNA and some of the limitations of the mitochondrial genome. So here's a figure of the mitochondrial genome. It's circular, it's small, it's found in multiple copies in nearly every cell of a body, and we inherit it from our mother. So all of us here have inherited our mitochondrial DNA from our mother, and so it traces the maternal line, or your mother's 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 line. And so here's something that demonstrates that, a figure that I put together. Squares are males, circles are females, horizontal lines are marriage, let's say, and vertical lines are offspring. And so if this is you, your mitochondrial DNA traces your mother, or your mother's mother, or your mother's mother, right, all the way along this line. It doesn't give you any information about all of these other ancestors that you have. So if you go back 3 generations, it's tracing 1 individual out of 8. If you go back 5 generations, it's tracing 1 individual out of 32, and so on. So it is limited in that way. So over the past decade, there have been rapid advances in genomic technologies and genomic analyses, whereas now, these days, instead of analyzing a small portion of the mitochondrial genome, maybe a small section of about 300 to 600 base pairs, we can now analyze the complete mitochondrial genome, all 16,500 base pairs. Or for the nuclear genome, whereas before these rapid advances, it was very difficult to get any nuclear DNA information, especially in ancestral ancient skeletal remains. We can now gain information from all 23 pairs of chromosomes of ancient individuals as well as living individuals through potentially saliva DNA analysis. And so that gives us information on all ancestors, not just through your maternal line. So we were able to use these genomic technologies in another partnership on the coast. So in 2009, Dr. Sobalski and I began a partnership with Tsimshian-speaking First Nations in the Prince Rupert Harbor region on genomic analyses of living community members and ancient ancestral human remains recovered from this area. Skeletal remains of over 250 individuals dating to between 500 and 6,000 years before present are currently housed at the Canadian Museum of History, which used to be the Canadian Museum of Civilization, in Gatineau, near Ottawa. And so when we partnered with this communities, it was clear that genetic or genomic information was going to be used in combination with other sources of information because they're a rich traditional knowledge base and oral histories, as well as archaeology in this region. So the First Nations allowed taking samples of the skeletal remains for limited destructive analysis. So we employed the same partnership principles as we did with the First Nations on the interior, but then we also added to some of these protocols. So to clarify the roles and expectations of the parties involved in the partnership, and to better safeguard the First Nations from potentially harmful results, we signed a contract with the First Nation communities. At the request of the communities, we expanded our communications to include their legal representatives, and we had, I can't tell you how many hours I've spent on the phone talking to lawyers and going down possible scenarios of findings, and it reminded me of some of the comments that Deborah had in her talk about exploring the possibilities of what could happen in the future. As well as we talked with community leadership and the general members of the communities. We continued to visit the communities in person annually, but in 2012 I had a major life-changing event. It's so extreme that I can barely remember the first year after this event. I had twins that just turned 3 last week. And so as a result of this, I wasn't able to travel up here as much as I would like. So I started relying on communication technology advances like Skype and other video conferencing to talk with the First Nation communities and the members of these communities. And what ended up happening is I ended up talking with them a lot more than I had recently, where I relied on my annual trips, in-person trips. And so that was ended up being a nice addition to working with these communities. And lastly, we invited community members to participate in a week-long workshop to learn the uses and limitations of genomic research called the SING Workshop. I want to discuss this workshop in detail, but before I do that, I'd just like to summarize some of the genomic results from our partnership on the coast, and Deborah has already kind of done my job for me, so I'll just go quickly through this. So we found, by looking at mitogenomes in mid-Holocene individuals as well as living community members, we were able to find these DNA affinities or links. So the first one that Deborah talked about was an individual from Lucy Island that we're referring to as 938, who radiocarbon dated to about 5,000 years ago, had a mitogenome that was very similar to another mitogenome from an individual who radiocarbon dated to 2,500 years ago, but was found on Dodge Island. That's near Prince Rupert. That's in the Inner Harbor. This individual had the same exact mitogenome as the Tsimshian-speaking person. We found another connection, but this was with multiple individuals from multiple communities in this geographic region. And then the last connection that I want to talk about was unexpected, where, again, from Lucy Island, an individual that we're referring to as 939, radiocarbon dated to around 6,000 years ago, and we didn't find any connections with living community members in this region, but we did find a close connection to a mitochondrial genome from another ancient individual from Prince of Wales Island, who dates to about 10,000 years ago. So we were able to find this mitogenomic connection from 10,000 years to 6,000 years, but not to living communities today. But we have to keep in mind that we're focusing here on the mitochondrial genome. And so we're just tracing this one ancestral line. So last year we were able to publish a study where we were actually able to look at a low coverage genome of this 939, of this individual that we're calling 939. And so this slide ended up being complicated, so I'm just going to break it down for you. So let's focus here on this map, and Deborah kind of talked about these types of analyses called F3 heat maps, and so it's kind of hard to see too. But the more the more red a circle is, these are community living present day communities, and the X is where the 939 individual is located. The more red a circle is, the greater genetic affinity to the 939 individual. And so you can see that in the Americas it's mostly red, right? But I think this figure kind of shows the genetic affinity with individual or communities in the same geographic region. So the farther to the right, the closer the genetic affinity to the 939, and clearly you can't see all of these names of all these communities, so I blew it up right here, and the close